Hello friends, welcome to the series of lectures of immunology. This is the 14th lecture and it will going to deal with the complement system. Complement refers to a group of large thermolabile enzymatic proteins found in the serum and body fluids. It completes the antigen-antibody reactions. It was discovered by Julius Bordet in 1890 and he named it as alixin. The term complement was coined by Paul Ehrlich. The complement system is a set of protein that acts together to destroy invading microorganism. Bordet devised a simple test for the lytic activity and that easily detects lysis of antibody coated red blood cells called hemolysis. Now let's talk about the salient features of complement. Complement is a collective term for a series of complex proteins found in normal serum. They are beta globulins. They are neither antigen nor antibodies. They exist as proenzymes and circulate as inactive form. On activation, they become active, acquire enzymatic or esterase activity. The activated complement is shown by putting a bar over them, whereas the inactive form of complement are indicated by a suffix I with them. They are thermolabile and are destroyed on exposure to room temperature or to a temperature of 56 degrees Celsius. The complement system is formed of about 20 different types of protein called beta globulins. Of these, about 16 are biologically important. Complement proteins are designated by the letter C and are numbered from C1 to C9. Complement C1 consists of 3 subunits, namely C1q, C1r and C1s. The complement proteins are capable of reacting with each other in a particular sequence as following. C1q is followed by C1r, which is followed by C1s, then C4, C3, C5, C6, C7, C8 and then finally C9. Complement performs a number of protective functions such as it brings about bacteriolysis and hemolysis in cooperation with antigen complex. It helps in phagocytosis of microorganism by phagocytes. It detoxifies the endotoxins of microbes. It brings about the coagulation of blood by converting prothrombin to thrombin. During complement activation, a complement component may be cleaved into two dissimilar fragments. The larger fragments usually joins the cascade. The smallest fragment usually possesses biological effects which contribute to defense mechanism by amplifying the inflammatory process, increasing vascular permeability, inducing smooth muscle contraction, causing chemotaxis of leukocytes, promoting viral neutralization, detoxifying endotoxins and affecting the release of histamine from mast cells. The complement proteins remain in an active form in the blood plasma. The complement are made active by certain substances called activating substances or activating agents. They include antigen-antibody complexes, gram-negative bacteria, animal viruses, aggregated antibodies like IgG or IgA, endotoxins, yeast, etc. When one component is activated, other components are triggered in a sequence, in a cascade pattern to bring about a biological activity such as lysis, phagocytosis, etc. Now, there are some unique properties of activated complement. When a complement is activated, it acquires the following unique properties. The activated complement binds onto the biological membrane such as the membrane of a bacterium. It generates enzyme activity. It activates the next complement protein in order to participate in a chain reaction. Some of the components of chain reaction becomes bound to the activating agents such as microbes. As a result, the structural configuration or the functional properties of the activating agents are altered. When the complement system is activated, it passes through two distinct phases or pathways. First is the classical pathway and second is the alternative pathway. Now let's talk about the first pathway, that is a classical pathway. Classical pathway is simple Weiss reaction brought about by activation of complement system. The classical pathway is activated by antigen-antibody complex. gram-negative bacteria and animal viruses. The complement components involved in the classical pathway are 11 components, namely from C1 and C9. The component C1 has 3 subunits, namely C1q, C1r and C1s. The 11 components of classical pathway react in the following sequence. C1q is followed by C1r, C1r is followed by C1s, which is followed by C4, C2, C3, C5, C6, C7, C8 and C9. The activating agents such as antigen antibody complex interacts with the first component of the complement C1. The C1q of complement C1 actually recognizes the antigen antibody complex and binds with the Fc portion of the antibody. This process of binding of complement to the antibody is called complement fixation. Calcium is essential for effective C1q binding. The attachment of C1q to the immune complex activates the C1r. Activated C1r function as serine histidine esterase and it activates C1s. C1s activates the complement C4 and C2 in the presence of magnesium ions. The complement C4 on activation splits into a smaller fragment called C4a and a larger fragment called C4b. The C4b becomes activated and the activated C4b becomes attached either to antibody C1 complex to the surface of the microbe. The activated C2 is also split into the small fragment called C2a and the larger fragment called C2b. C2b reacts with C4b and forms a complex C4b-2b complex. This C4b-2b complex function as C3 convertase enzyme and it activates C3 component. The activated C3 component is cleaved into a small fragment C3a and a large fragment C3b. The C3b binds with C4b-2b complex to form C4b-2b-3b complex. The 3b has binding site for macrophage and phagocytes and hence the C4b-2b-3b complex is adhered to macrophage or phagocytes along with the microbes. Now let us take care of functions of C3b. The C3b has the following functions. It facilitates the adherence of bacteria and viruses to macrophages and phagocytes. It facilitates the ingestion of certain bacteria by macrophage and phagocytes. It augments IgG-induced phagocytosis and IgG-mediated cytotoxicity. The C4b-2b-3b complex activates the next complement C5. The activated C5 splits into a small fragment called C5a and a large fragment called C5b. The C5b has two combining sites. One site is to bind with antigen, that is microbe, and the other site is to bind with complement factor C6 and C7. C5b binds with C6 and C7 to form a trimolecular complex called C5b-C7 complex. It can remain in the solution or bind to the surface of the microbe. When it is bound to the microbe, it is stable. When it is free in solution, C5b-C7 has a half-life of only 0.1 second. C5b-6-7 complex binds with C8 to form tetramolecular complex called C5b-C6-7-8 complex. The C5b-C6-7-8 complex has a binding site for C9. It binds with C9 to form C5b-C6-7-8-9 complex. This complex causes the disruption of the lipid layer of the membrane of the microbe. As a result, a hole is made on the microbe. Through this hole, the contents of the cells are released and the cell is lysed. This phenomena is called bacteriolysis. When the lysed cell is an RBC, the lysis is called hemolysis. Now let us talk about the significance of the classical pathway. When a microbe enters the body responds by producing antibody. The antibody binds with the microbe to form an antigen antibody complex or the immune complex. When the immune complex is formed, the complement system is activated to enhance the removal of the microbe. This is done in the following ways. The complement system helps to adhere the microbes to the neutrophils and macrophages. It facilitates the ingestion of microbes by neutrophils and macrophages through phagocytosis. It brings about the lysis of the microbes. It augments the IgG mediated cell cytotoxicity. Now let us talk about the another pathway of complement fixation, that is known as the alternative pathway. It is also known as Properdin pathway. The activation of complement C3 without the prior participation of C1, C4 and C2 is called alternative pathway. The alternative pathway was discovered by Pelliner in 1954. The alternative pathway is a stepwise reaction of complement system to bring about the destruction of microbe in the body. The alternative pathway is activated by aggregated antibodies IgG or IgA, lipopolysaccharide, bacterial endotoxins and yeast. The alternative pathway of the independent of Fc fragment of the antibodies. It means that this pathway is independent of the Fc fragment of antibody. About 6 complement components are involved in the alternative pathway. They are C3, B, D, P, H and I. The alternative pathway does not involve the components C1, C4 and C2. In this pathway, C3 is the starting component and is activated without the activation of C1, C4 and C2. In alternative pathway, C3 is activated by PZ complex. PZ complex is formed by the reaction of Properdin, a normal serum protein, with zymosin, which is a polysaccharide from yeast cell wall, in the presence of magnesium ion. The zymosin of yeast function as the activating agent. The PZ complex function as a proteinase enzyme and it cleaves C3 into a small fragment C3a and a large fragment C3b. C3b is coated on the surface of the microbe. Then factor B binds with C3b to form a complex called C3b-B complex. The C3b-B complex is a weak bridge and is stabilized by the factor P called Properdin. Activated factor D activates and cleaves the C3b-B complex, releasing C3 and cleavage enzyme C3b-B called C3 convertase. The activated C3b-B splits C3 into C3a and C3b. C3b binds to form. C3b formed can be used in several ways. It can be deposited on the nearby microbe and interacts with the factor B and D. Once C3b is formed, a cycle of activation occurs which maintains C3b formation without the original activating agents. The C3b may activate the C5 and enter classical pathway and produces C5b 6, 7, 8, 9 complex in sequence, resulting in the lysis of the microbe. Now let us talk about the biological functions of complement system, which includes immune adherence, chemotaxis, opsonization, phagocytosis, lysis and coagulation. Let us talk about each point one by one. Now let us talk about each biological function step by step. First is immune adherence. The complement component facilitates the binding of microbe to the phagocytic cells. This is possible because of phagocytic cells have receptors for C3b component and the microbes coated with C3b are adhered to phagocytes. Is chemotaxis. Complement bound antigen antibody complexes release chemotactic factors and attract leukocytes, which in turn releases lysosomal enzymes. This helps in the localization and inactivation of infectious agent or may enhance tissue injury. The third function is opsonization. Opsonization is the rendering of bacteria and other cells to phagocytosis. During opsonization, the complement components bind with antigen antibody complex. The complement bound to antigen antibody complex has a binding site to phagocytes. When the phagocytes is bound to the complement, the phagocytosis is facilitated. Fourth biological function is phagocytosis. Complement system facilitates ingestion of microbes by macrophages and neutrophils. And the fifth biological function is lysis, that is the attachment of complement factors to the cell membrane of the microbes causes disruption of the lipid bilayer of the membrane of the microbe. As a result, a hole is made on the microbe. Through the hole, the contents of the cells are released and the cell dies. This is called lysis. When a body cell such as tumor cells is lysed, the lysis is called cytolysis. If a bacterial cell is lysed, the lysis is called bacteriolysis, and when a RBC is lysed, the lysis is called hemolysis. Now, the another reaction is anaphylactoid reaction. The complement C3A and C5A are considered as anaphylotoxins. They causes the release of histamine from mast cells. They have kinin-like activity and also chemotactic activity. The clinical symptoms caused by anaphylatoxin of C3A and C5A are exactly similar to that of anaphylactic reaction of type 1 hypersensitivity. The type 1 hypersensitivity is specific immunological reaction, but the complement triggered reactions are non-specific. This non-specific clinical reactions produced by complement components are called anaphylactoid reactions. Complement system brings about the coagulation of blood. The blood platelets adheres to activated C3 are lysed. The lysed platelets release a factor which converts prothrombin into thrombin. Thrombin brings about coagulation. Now, these are the applications of complement fixation, which include Weissen's test. It is a complement fixation. When the complement fixation is applied to diagnose syphilis, it is known as Weissen's test, and it is also help in detection of viral infection, in which complement test is used to identify viral infection and rickettsias. These are the references that you can go for your further studies. Hope you are going to find this lecture on complement system beneficial for your understanding, to understand the various aspects of immunology and various other reactions undergoing in this system. Thank you.